Alright guys, welcome back to this slowly but surely progressing biochem molecular session. Once this entire playlist of the whole first aid is done, the teachings that it'll bring to future students is pretty unmatched. As usual, feel free to shoot me an email if you're trying to get your hands on my annotated first aid, if you want to use that for yourself. And I also do offer a Step 1 as well as a Step 2 CK course, so if you're interested in either one of those, you can shoot me an email and I'll give you the details on that. Last time we left off at a topic of DNA replication, which in itself is very intense, and there's a lot of enzymes and what they do, and we touched base on all of that. Now we're moving on to mutations in DNA, which is the next page on the biochem molecular section. And we have right here a little image that gives you a basic understanding of what happens in mutations by utilizing a basic English sentence. DNA has all you can ask for. So you can see the sentence used 3-letter words, just like codons, and that first sentence is your normal DNA, basically. Then you can have 3 different types of what they call a point mutation: silent, missense, or nonsense. Those are all point mutations which occur at a specific locus, and that's the only point where the mutation exists. All right. So that's going to be different from a frameshift mutation, where you can see that the locus of the mutation highlighted in yellow is that Y, but then everything downwards is also practically mutated, right? So it wasn't just at one point. The error doesn't exist at just one point, which makes frameshift mutations pretty much impossible to repair, right? So that's the key point to keep in mind right there. They also mention that point mutations can either be transition mutations or transversion mutations. So transition is just transitioning from one to another of the same type, right? So you can go from purine to purine, so A can become G, or pyrimidine to pyrimidine, where C can become T, right? But the type stayed the same, which is key because purines are smaller, pyrimidines are bigger. So when you replace a purine with a pyrimidine by mistake, that actually makes a little clunk in the chain of the DNA, and that's often something that can be caught very easily, and it leads to future issues when they try to replicate that same DNA again. A transversion is a conversion, right? So you converted a purine to a pyrimidine, or a pyrimidine to a purine. So this is where you're going to have that problem where you now are going to notice that clunk because your purine, which is smaller, became a pyrimidine, right? So having a transition mutation usually is okay. It's something that might not cause a clunk and will not lead to issues other than the general aspect of changing the letter in your sentence. But a transversion mutation is more problematic because now you have that little clunk since you replace a purine with a pyrimidine, or vice versa, and that size difference plays a major dynamic in how your DNA is going to unfold, how other enzymes are going to attach to it, and etc. That being said, we can dive into each one of these in itself. So silent mutations are nucleotide substitutions that code for the same amino acid, usually thanks to the wobble aspect of tRNA wobble that we talked about in our last session. So that way, nothing really changes, hence it being a silent mutation. So if you read that sentence of the silent mutation, it still reads out the same way as a normal sentence. DNA has all you can ask for, but the U is now spelled incorrectly, but it's still functionally doing its job in the sentence. Missense mutations are where you take a nucleotide substitution resulting in changed amino acids, right? So now you have a different word there. This missense mutation could be a conservative missense mutation if the new amino acid is very similar in chemical structure to the original that was there, in which case it'll act pretty similar to a silent mutation, right? Otherwise, if you completely change the amino acid to something completely different chemically, like an acidic one becoming a basic one, or something with a low pH becoming a high pH one, and so on, or even the charge of some of these amino acids, right? You might take something that's neutral and replace it with a negative amino acid, whatever. Those things can cause issues in the tertiary structure when these amino acids come to be, and they're all going to bend and form a quaternary structure in the very end. They're not going to be able to fold correctly if the pH or the charge is incorrect based off of the original blueprint that it was supposed to have. This is very notorious in sickle cell, where you replace glutamic acid, which is acidic pH, with valine, right? So that's going to be a problem now because that charge and that pH differential now won't allow the new protein to fold correctly, and hence the problems that occur in sickle cell. A nonsense mutation, as you can see in the image right there, DNA has all U. See, the point of the mutation is at the word can, and then that's it. So that mutation itself is actually the insertion of a stop codon, UAG, UAA, or UGA, which you have to memorize, And once the stop codon gets inserted, you can already tell that the new protein you're going to make is going to be significantly smaller than what you were supposed to make, right? And the last one here, a frame shift mutation, which is not a a point mutation. This is a deletion or insertion of a number of nucleotides not divisible by 3. So either 1 or 2 or some other multiple of 1 or 2 that isn't divisible by 3 has now been inserted or deleted, resulting in misreading of all the nucleotides downstream from there on. So that sentence is no longer even readable anymore, right? DNA has ALE and then ONC, ANA, SKF, OR. And the last guy isn't even a full codon. It's just OR, it's 2 letters. So this is really problematic because now you get an entire protein that's completely messed up. The problem here can be many different variations, right? You can have many different variations of a frame shift mutation, because you could have a frame shift mutation where you're going to make 1, 2, 3, 4, 5, 6 things, and then the 7th one won't actually give you an amino acid. So you'll make a new protein that's encompassed by these 6 amino acids, of which the last 4 are not even the correct amino acids. So you do get some protein here, but now you have to worry, because is this protein functional? Is this protein non-functional? Is this protein going to function to do something really bad, right? You can make a malignant protein of some sort that's going to cause new issues now, right? That's really unfortunate. Of course, a frame shift mutation could also occur in such a way that you ended up creating a stop codon, right? So that's another thing you have to watch out for, to not get confused between frame shift versus nonsense mutations. You could have a deletion of 1 or 2 or insertion of 1 or 2 amino acids in such a way that you now created a stop codon, which of course will yield a smaller protein, but what you have to look for is what happened to the rest of it. What happened to the rest of the everything else afterwards, because there are still going to be, you know, triplet codons here and here as well. They will be there, they just won't be read because you have now put a period right there, right? So you need to see what happened in the frame shift one, where you should still have, of course, the rest of your codons, and if they all look correct and it's now starting to look like something's weird, then that's a nonsense, right? Because this is still correct, this is still correct, right? So the only mutation is at one point, so that's when you call it a nonsense mutation. But if you did a frame shift mutation, and let's say maybe now this actually became a stop codon. Okay, so let's say this became a stop codon, very similar to a nonsense, right? You did a frame shift, and the word OB now became a stop codon. Is this a nonsense mutation or a frame shift mutation? Well, you look at the rest of your amino acids. When you look at the rest of the amino acids, they do not look correct at all. The entire thing has been messed up, so that's a frame shift mutation. That's a very key thing to make sure to be able to differentiate between the two. You will have all of these as answer choices. If you ever get a question on a mutation and you need to identify the mutation, they're all going to be answer choices. So the answer choices are never something that are going to help you, right? With principles, it's all about the knowledge, and really just nothing else but the knowledge. When it comes to organ system problems, it's about you being able to apply your knowledge in developing a suspicion for what you think might have been wrong with the patient. Developing a suspicion with the patient starts from the moment you lay your eyes on the patient, their age, how they look, whether they're well kept, if they're acting a certain way, the general physical exam, right? The labs you look at, all of these things are supposed to point at suspicions. You won't have those things when it comes to principle-based questions like biochem. So it's all really based off of knowledge, and that's the knowledge I'm trying to help you figure out here, so that you can truly know more about these guys than what's given to you in the book itself. On top of that, there's this other aspect of a Splicesite mutation. Right? So a Splicesite mutation is the different type of mutation, different from all of these, in the sense that the mutation now only occurs really at the splice site. The site of splicing will now lead to a retained intron in the mRNA, right? Because remember, the splice sites are there to let you know which introns you need to get rid of, right? Because exons are what get expressed, introns are intervening. If you don't get rid of the intron, it gets left in the mRNA, which could then lead to a protein that doesn't work, right? So that's pretty problematic. And splice site mutation is an answer choice you really only pick if you're getting an experimental question where some scientists are working on something, and it might be related to cancers, dementia, epilepsy, some types of beta thalassemia. All of those are splice site mutations. So you just pick that as an answer choice and you move forward, and you should be pretty confident in that, because you don't need to worry about picking one of those other things if they didn't give you a DNA sequence to really work with there. All right. And then I have a little note here from my book. Note that both nonsense and frameshift mutations can lead to a smaller protein. However, only frameshift may actually result in a longer protein by changing a stop codon. So that's the third case scenario that can occur with a frameshift. Instead of creating a stop codon, what you could have done is you could have actually had, let me not do that. You could have actually had a frameshift that got rid of the stop codon. So now, where you were supposed to have a stop codon is no longer a stop codon. So frameshift mutations could actually lead to a protein that's longer than what you were originally supposed to have. Because remember, frameshift isn't just deletion. It could also be the insertion of a number of nucleotides not divisible by 3. Right? So frameshift is the only time you can get a longer protein. Nonsense and frameshift are both opportunities where you could get a smaller protein, right, depending on what occurred in that frameshift. So these are some key things to watch out for, to use as hints for yourself when you're tackling questions where something weird happened. All right. I think earlier we already talked about did we already talk about Lac Operon? No, we haven't talked about Lac. Okay, so next topic here is your Lac Operon. So let's dive into that. That's another topic that a lot of students tend to get pretty confused about. There are other operons, so I guess that's, the reason I'm telling you that is because you should be grateful you don't need to know them. There are many other operons that you could have to know as well, but here we're only really working with the Lac Operon for step 1. So there's a couple things involved with the Lac Operon, and you can just look at the notes in your book for this, right? So this is a classic example of a genetic response to an environmental change. In our bodies, all of us are always having genetic responses to something that changed in our environment. This is kind of what plays a big role in the aspect of survival of the fittest, right? Some species in the past may not have been able to adapt to changes that occurred in their environment, and then they become extinct, right? So here you see glucose is the preferred metabolic substrate in E. coli. That's what most bacteria like anyway. And the Lac Operon is one of the major aspects of E. coli function. When glucose is absent, remember that E. coli is also a lactose fermenter. So E. coli can use lactose if lactose is available. The Lac Operon will be activated to switch to lactose metabolism. That's what the Lac Operon does. That's why it's called the Lac Operon. It operates in helping you use lactose. So it goes to say that if you have a lot of glucose or if you have no lactose, either one of those two case scenarios, high glucose or low lactose, your Lac Operon will not be working. There's no reason to, right? Because if you have a lot of glucose, you're just going to use that. You don't need your Lac Operon. And if you have no lactose, then you can't use your Lac Operon, so it won't work, right? So those are two things that are case scenarios where your Lac Operon will not work. So when will it work? There's only one case scenario where the Lac Operon will have full functionality. That's going to be the case scenario where you have low glucose and you have high lactose, right? Low glucose, high lactose. That way, E. coli will realize there's no glucose for it to use, but then it'll realize there is some lactose it could use, so it'll turn on the Lac Operon. Right? Now, how does the system work to make sure that this is kept in check? Well, there's two ways to do it, two distinct regulatory mechanisms here. The first one is negative regulation. It's negatively regulated by binding of the repressor protein to the operator locus when lactose is missing. So that you can see there's something called the repressor protein where once allolactose binds to the repressor protein will not work anymore, thereby allowing the Lac Operon to do its job, right? So let's add that in here. Negative regulation with Lac, they call it the Lac L, well, lowercase l, repressor protein, right? This repressor protein will usually be on the operator itself, stopping it from working. By being on the operator, it prevents the transcription of all the main genes that need to be transcribed to make whatever proteins you need to utilize lactose, right? So that's your negative regulation. Then there's some positive regulation as well. Positive regulation occurs with the aspect of cyclic AMP and the CAP protein. So this guy, the CAP, that CAP site is right before the promoter, as you can see in the picture on your book. The CAP site is actually supposed to act as something that upregulates the promoter, thereby allowing more transcription to occur. Now, these two guys work with each other. So if the promoter is working, but the operator comes next, and the operator is blocked off, then the promoter can't really do much, right? And it's going to be minimal expression. You have to keep in mind that the same DNA sequence you're looking at in your book, there are more than just one, right? There's like, let's assume this is a colony of 1 million E. coli. All of them are doing this. But in some of them, the operator might not be working so well, and the promoter might be working, so they might have minimal presentation and actual function of those LAC genes, which are going to make the lactose utilizing proteins, right? But overall, if the operator is on the repressor protein is on the operator, then you're not really going to be doing any transcription of your LAC genes. So these guys work in that order, right? First comes the promoter, then the operator, right? So the promoter is going to go tell you to do the transcription, but then the operator has to basically approve of that, and the guy stopping the operator is your repressor protein. So everything needs to be approved together for you to successfully go ahead and do transcription of your LAC genes. There's Z, Y, and A, 3 of them, right? This is considered a polycistronic mRNA. Very key word to keep in mind. It's not really in the book. This basically means that this is one round of transcription that will encompass all 3 genes onto the same mRNA, right? You're not transcribing each one separately and creating a separate mRNA for each one. This is something that bacteria are able to do, and prokaryotes are very good at it, right? Not entirely sure if eukaryotes do it, but prokaryotes are definitely very good at it. So now you could see the case scenario. This is something you have to know for biochem metabolism as well. When glucose is present, adenylate cyclase will not work. Adenylate cyclase is an enzyme that only works when glucose is not present. So, in the absence of glucose, adenylate cyclase will work. Oh, what happened there? Absence of glucose equals adenylate cyclase activity, right? Now adenylate cyclase will take ATP and convert it into cyclic AMP. This is pretty logical, right? When you have sugar, you think of that as energy, and energy is ATP. When you don't have glucose, right, absence of glucose, you're not going to have ATP anymore. Now you're going to have cyclic AMP. Okay, cyclic AMP upregulates the CAP, right? So now the CAP protein can go to the CAP site and then upregulate your promoter. Boom, so far so good. Now, the promoter is one of those sequences that allows more transcription to occur of the main genes we're looking at, the LAC, Z, Y, and A. No K over here. All right, so now, for the promoter to succeed, you would need the repressor guy to not be there, right? So promoter now needs approval by the operator. Okay, now on the operator is blocked by repressor unless lactose is present. Right, and that's the story here. So at the bottom half of that genetic image they're showing you, you can see that lactose is actually able to go block off the repressor protein from doing its job. So now repressor protein is not working. So in the absence of glucose, promoter was working, and in the presence of lactose, the operator will approve transcription. And that's it. That's really the only thing you need to know about here. It's really not as complex as the book kind of makes it look like. I mean, the images they show on the right side with the 4 different states and what occurs in each of them is really just there to break it down for you. So if you look at each of the states, the first state says low glucose, lactose is available. So if you have low glucose, then of course adenylate cyclase will be working and you're going to be successfully making the CAP. And if lactose is available, then that's great. That means that the repressor protein will no longer be functioning. And this is the case scenario you'll get where your operator will approve transcription, and boom, you're going to express all your LAC genes and you're going to make the LAC ZYA proteins to go ahead and utilize lactose. So that's the case scenario that's not the ideal case scenario, but the ideal case scenario when glucose is not available. In the second scenario, you see high glucose, lactose unavailable. That's a pretty ideal case scenario because if you have a lot of glucose, E. coli is very happy, everything's great. Right? So you can see that the repressor protein is sitting right there blocking the operator, right, in that second case scenario. And since you have a lot of glucose, you have a lot of ATP, no CAP function, no cyclic AMP and CAP function. The third case scenario is where you have low glucose and low or lactose unavailable, low lactose, right? So now that is the least ideal case scenario because now this E. coli colony will have no way to survive. Since there's low glucose, the CAP site will be there, the cyclic AMP will be working, but since there's no lactose, the repressor protein will not approve the operator, right? So then there's no approval of transcription and nothing will be made. And this E. coli will basically die. The last case scenario is where you have everything, abundance, right? So you have high glucose and high lactose. So then, of course, when you have high glucose, the CAP will not be working. There's no cyclic AMP because when you have glucose, adenylate cyclase doesn't work and you're just full of ATP. And that's not, what you're going to learn in this fourth scenario now is that's not necessary for you to transcribe your lac proteins, okay? What is more necessary for you to actually transcribe your lac proteins is for the operator to approve. The operator needs to approve. A promoter, remember that in genetics, a promoter gene doesn't need to be functioning for you to do transcription. A promoter just upregulates the same transcription that you were going to do anyway. You'll just do it faster. So now in the fourth scenario, you can see that since lactose is also available, because this is the scenario of abundance, the repressor protein is not working. So the operator is open to approve, and you approve expression of your lactose genes. However, in this fourth case scenario, since you're already utilizing glucose, you probably won't need too much of it. So there's going to be minimal, or as they say, very low basal expression of the lactose genes. So here there was a couple things to understand. There was the negative and positive regulation to understand and how it works, and then the group work of how the whole system works together to respond to what's available in the environment. All right. So that takes care of your topic of mutations on DNA and notorious Lac Operon. In the next video, we'll go on to talk about DNA repair and how the different types of DNA repairs allow for you to survive everyday damage that your DNA takes. And we'll move on from there to the other topics regarding DNA management.